Okay, good afternoon, everybody. Welcome to Microbiology 3300. My name is David Muir. I'm going to be your teacher for the rest of the semester. Let me just explain. I know that this is not, for many of you, the ideal situation, having virtual classes. Nonetheless, there are huge advantages to having recorded classes like this. And that is that you can do this class at your own time. You can repeat the class over and over again. And that is the key to acquisition of new knowledge, new information, is repetition. I strongly advise that the PowerPoints for each class will go up prior to the class, and that you go through the PowerPoints, at least briefly, before reviewing this Zoom presentation, which will be presented for each class. You can view it several times, make notes every time, make notes of notes, make summaries of your notes, make notes about your summaries. The more you repeat, the more you push stuff through new neural pathways, the better you will acquire the knowledge. The more the knowledge will become yours. There will be no struggle to learn if you do it by repetition like that. It is no good leaving things till the very last minute. It does put a tremendous burden of responsibility on you to keep up with these classes. Do not let the classes stack up, especially because each Wednesday, for example, you have a problem set on mastering microbiology, which you're going to have to complete. And those two tend to pile up if you don't stick to them, stick with them. Okay, so keep up, set a routine, come here at the class time and view the class. Hopefully it'll be up on time every time, and so make sure that you set aside the time to view it. Okay, please go to your syllabus. I'm not going to go over the syllabus. Please go to the syllabus, read through it carefully, and make note of the rules and regulations, and also have a look at the calendar. We'll try to stick to the calendar as much as possible. In addition, please note the way in which grading will be constructed, and realize, please, that at the end of the semester, the grade you get is a numerical computation. It's not a decision on my part about how I feel about you. It is something that depends on how well you've done during the semester. So please keep up with things and be aware of how you are doing all the way through. Okay, so this is microbiology. Microbiology is the study of very small things. Formally, microbiology is the study of things which are too small for the human eye to resolve. And the limit of resolution of the human eye, the smallest that we can see, is 0.1 millimeters. So 0.1 millimeters is already very small, but the microbial world goes much, much smaller than that, very much smaller than that. So other thing to realize is we are surrounded by microbes. There are microbes everywhere, absolutely everywhere, on every surface, in every fluid, in the sea, in the mountains, in the plant, everywhere. There are a gazillion kinds of microbes, and they are all invisible to us. They're all invisible unless we use some form of magnification. And this is very important to understand, that the very first steps in the science of microbiology were made when we acquired the ability to see these very small things. I should explain that interest in the natural world has been part of mankind's history since whenever. But modern science, as we understand it, the modern bodies of knowledge as well, began to be laid down really in about the 1600s. And one of the motivations for that was that wealthy gentlemen with leisure and money liked to become natural historians. They liked to examine the world around them. Many of them set up little museums. It became a thing where people had their own little museum. They called it a cabinet of curiosities. They would have fossils and eggs and stuffed birds and whatever. And periodically, gentlemen would get together and discuss their interests and then go off and pursue those interests. And then they began to formalize these kind of gatherings and everything into society, into scientific societies, and began to take a very important step which characterizes modern science. And that is that they would publish. They would publish their results, and other people would be able to examine the results, criticize them, act on them, repeat them, and elaborate on them. That's how science works. As a scientist, you always expect to see your work demolished eventually by succeeding work, which builds on it, doesn't destroy it. It rebuilds on your work. These were its origins, these kind of societies. So in London, there was one gentleman called Robert Hooke, 1665, who, along with a number of other of these gentlemen, began to make lenses which were able to magnify quite highly. Lenses had been a part of human history for a very long time. Even the Romans had reading glasses. And they learned that if you could make a lens which was very spherical, the more spherical the lens became, the more it would magnify. But there was a big problem, because the more round you make it, the shorter the focal length, the closer to the lens you have to look, and there's a limit. So Robert Hooke actually, his lenses were just magnifying glasses. They were just good magnifying glasses. That's all they were. But he was able to make one very important discovery with that magnifying glass. And that was, he looked at cork. He took a wine bottle cork, shaved it very, very thin, and examined it with his microscope. And what he saw were little boxes. They looked like little boxes all stacked together. And these were not living cells, obviously. This was not living cork. It's dead. But those are the cork, those little boxes are the cell walls of cells which had since died. But he described them, and he drew them, and he called them, so he was the first person to use that word cell. And that was a huge breakthrough, a huge starting off point for biologists, because we in fact now recognize that the cell characterizes life on our planet. All life is cellular in nature. Hooke was the first person to describe cells. He later on described living cells as well, because he looked at fungi, and he described those living cells of fungi. This was the beginning of cell theory. All living things are composed of cells. There's another part to cell theory which is equally important, which we're going to repeat in a minute. And that is that all cells arise from another cell. All cells arise from another cell. There was one event when a cell evolved, and all other cells have evolved from that original cell. We don't get new cells arising. So that was Robert Hooke. It sounds like a simple thing to have done, but it was a revolutionary idea at the time. Now, the next really important person to talk about is this gentleman here. This is Anton von Leeuwenhoek. He was a Dutchman. He was a rich merchant. He was wealthy, had leisure time, and an obsession with developing lenses, with making lenses. And he was very, very, very good at it. And he could make very good, almost spherical lenses. They had to be very small, though. And because they were small and because their focal length was so short, you couldn't like hold them to your eye and look at things like that. Instead, he invented a little machine, and here it is here. He had a plate and a hole in it where he could put the lens, the spherical lens. Behind it, there was a pin, and on that pin, he could put a specimen. And it had a screw which moved up and down, so he could get it into position to see it. But then his absolutely revolutionary breakthrough was he made a focusing screw. He could move that pin back and forth until the specimen came into focus. And with a good lens and with good focus, it was absolutely astonishing what he could see. He was the very first person to see bacteria, for example. And not only did he see bacteria, he could see them clearly, and he described them. This was the other thing that was remarkable about Lutheran Hook. He was a very good artist, a biological artist, and he drew everything that he saw, and he communicated. He communicated especially with the Royal Society in London, the world's most prominent scientists. And he sent a flood of information to them throughout his life. Here are bacteria that he saw. He was the first to distinguish different bacterial shapes. Here you can see a coccus. Here you can see cocci arranged. This is called a palisade. These are rods here. And he was the first person to describe bacterial motion. That bacteria move very well. A lot of them are highly motile. He was the first person to describe that. He also described spirilla bacteria. And then he described many protozoa. He loved looking at water. And he described many protozoa that occur in water. He did all sorts of things. There was no fluid which Van Leeuwenhoek did not examine, including diarrhea. That was one, and he made a very important discovery looking at his own diarrhea. And that was that he saw in his diarrhea, he saw a protozoan called Giardia. And his drawings are absolutely extraordinary because Giardia is very difficult to see, even with a good microscope. It's difficult to see all of its structure. He had it perfectly. He also was the first to describe his own sperm. He drew it and he described how it worked. He described how the sperm had a head and a long tail, and he could even see the nucleus inside the head of the sperm. This gave rise, I should tell you, to a rather loopy idea about human reproduction, in which the people seeing the drawings of the sperm and then confirming its structure, other people did the same, repeated it and confirmed the structure. Seeing the nucleus, they thought it was a little tiny baby, all folded up inside the sperm, and that the sperm then traveled up to the uterus and then stuck it into the uterus, and then this little tiny baby, which they call a homunculus, would then grow into the baby. But of course, that's completely wrong. But it seemed logical to them. Anyway, Van Leeuwenhoek really described the microbial world from bacteria on up extremely well. It was a remarkable achievement, and it opened up the study of these organisms. He really opened the idea that there was this whole world which needed to be studied. But sitting on it, sitting over the science, was this terrible theory of spontaneous, it was called spontaneous, the theory of spontaneous generation. Van Leeuwenhoek called all these little things that he saw, he called them animalcules, little animals. And many people believed that these little animals arose spontaneously, that you could take pure water, for example, set it aside, and it would spontaneously generate these little animalcules. People who who believed that, were called the vitalists. They were called vitalists because they thought there's a vital force that permeates the whole globe. And wherever it gets a chance, wherever the right conditions are present, it forces life into being. That's what they thought. It was a very, very dangerous theory because any experiment that you performed, the results could always be explained away by saying, oh, well, spontaneous generation led to the formation of organisms that did this. So it was very, very, very bad for science. And it was stultifying for science for centuries. There was an opposing viewpoint, and these were called biogenicists. And they believed that this did not occur, that in order to produce a cell of any sort, it had to arise from another cell. So it arose from pre-existing cells. Van Leeuwenhoek was one of these. And he was quite advanced for his time in that way because, for example, most people believed that maggots, fly maggots and flies arose from rotting meat. That if you let meat rot, it would spontaneously generate maggots, which would mature into flies. Van Leeuwenhoek did not believe that. He watched meat very carefully. He saw flies actually laying eggs. He saw eggs hatching. And he described them. He said, this spontaneous generation is nonsense. These maggots come from fly eggs, which reproduce just like anything else. Flies come from flies. They don't come from rotting meat. But it's actually required careful experimentation and nearly 100 years of it to put the theory of spontaneous generation to rest, and believe it or not, there are still people who believe in it. But nearly 100 years of experimentation before definitive experiments showed that spontaneous generation did not occur. So here's one of the early experiments. This is Francesco Redi, who's an Italian, and this is the experiment he performed. And it looks to our eyes very simple, but it encompasses all of the features of a true scientific experiment. And that runs as follows. He took rotting meat. He worked with rotting meat. He took an open jar and he put meat in the bottom. He took another jar and he put rotting meat in it. He put meat in it to rot, and he covered it with gauze. And he left them. And of course, the rotting meat in the open jar, the flies came, laid eggs, and maggots infested it. Covered, no maggots. And he presented this as an argument, as an experiment to show that spontaneous generation did not occur, and then that people like Van Leeuwenhoek were correct. Flies needed to lay eggs for maggots to grow. It sounds so stupid to us. But remember that people, once they have a theory, once they have something set in their minds, it's very difficult to get them to change their minds, even with obvious facts. We see this in our lives around us today to the nth degree. Francesco Redi, it looks to us like his experiment shows that, at least in that instance, spontaneous generation does not occur. The vitalist said that his gauze here impeded the vital force. So it impeded the vital force, and that's why it didn't grow maggots. But if you didn't impede the vital force, it needed air contact without any barrier in order for it. You see how the theory can kill even a simple experiment, kill the conclusions even to a simple experiment. It's very, very dangerous. And despite what looks like an obvious result, as it was not obvious to many people, they carried on arguing about it, as I say, for another 100 years before it was finally laid to rest. People began doing all sorts of experiments to try and demonstrate it. Here's a man, is John Needham. His name is John Needham. And he was a vitalist. And so he did an experiment which he thought proved that this life force actually was a real thing. So what he did was he took a broth, like a soup broth, and he boiled it to kill everything that might have been living in it. And then he left it, and he just left it alone. And then after some time, that broth began to rot. And it began and he began to see scum and stuff. And when he looked at it with lens, he could see things moving around in it, bacteria. And he said, this proves that provided the vital force has access to the materials, the vital force will cause spontaneous generation of organisms. This is a very difficult thing to argue about. Okay, with limited knowledge, it's very difficult to say he's wrong. But other people said, no, this is absolute nonsense. So here's the next step. This is a man called Lazaro Palanciani, and he did repeated Needham in a way. He wanted to disprove Needham, and so he looked at his experiment and he said, no, there's a step which has been left out in this experiment. So he repeated Needham's experiment. He boiled broth, left it open. There was the growth. But he went a step further. He took broth, boiled it, and then he sealed the flask. And there was no growth. But when he took the lid off of the thing, growth would occur. And he said, it's not a vital force. There is something coming in, but it's not a vital force. Somehow or other, this broth becomes contaminated. There's some living thing comes in, which we can't see, which causes this. The vitalists, of course, said, no. You've stopped the vital force from coming in as soon as you let the vital force in. Spontaneous generation. You see how this theory always finds a way to be correct. It's very difficult now for these. Palanciani has his ideas. Needham has his ideas, and they fight with one another because there is missing knowledge that does not let them get a satisfactory answer. This man is very important. This is quite late. This is in the 1800s, 1858. This scientist, Virchow, he was the first to tell us that every cell that you see, whether it's bacterial cell or any other kind of cell, every cell arises from another pre-existing cell, as I told you. We all arise from one original cell, our ultimate ancestor, way back. So that was our ultimate ancestor. But Virchow said spontaneous generation does not occur. We never have new cells arising unless they come from an existing cell. It's a very profound difference in thinking. And it leads us ultimately to this idea that actually we all came from an original cell once upon a time, no matter what organism we are. So this argument went back and forth, back and forth, and back and forth, until this wonderful scientist, Louis Pasteur, look how late it is, by the way, 1861, okay. More than 100 years of fighting about this spontaneous generation. Louis Pasteur did many, many, many things. He's one of the world's greatest ever scientists, let alone microbiologists. Spontaneous generation theory said, as long as you had the raw materials like a broth, and the broth had free access to the air, to the vital force, life would spontaneously arise. So here's what Louis Pasteur did. He made special flasks. They had a long neck like this. Okay, so through the neck he would pour a broth. And that broth was probably already contaminated with microorganisms, but what he did was he would then take this and he'd bend the neck into this shape, this S shape, and he then boiled it very vigorously to kill the microorganisms that were present. And he then left it. And he left the flasks like this, and nothing happened. There was no growth, no matter how long he left it. In fact, there are still some flasks which Pasteur set up in 1861 at the Pasteur Institute in Paris, still sitting there with broth in them and no growth. They had free access of the vital force, free access of air, but they didn't grow. Why? Well, the answer is simple. It's because things like bacteria in the air and their spores, they are objects. No matter how small they are objects. They have weight and they tend to settle. And that is what happens when you have a flask of broth and you leave it open. The bacteria or their spores end up settling into the broth. With his swan neck flask, that's what they were called, this S shape was called a swan neck. That couldn't happen. Bacteria will certainly fall in, but they settle here. There's no air movement to carry them in to the interior of the flask, and it's too far anyway. But they tend to settle here, and no growth takes place as long as they're not contaminated by bacteria or by their bacterial spores. He took this another step further. He left these for a while, and then with some of them, he snapped the neck here, and then others, he tipped the flask over. He let some of the broth flow here to get contaminated, and then he turned them back upright again. And in those with broken flask, with broken neck, or where they were tipped like that, the bacteria grew. The ones which were left standing upright like this did not grow. And his explanation was very clear. In order for that nutrient to grow bacteria, it needed to be contaminated. It needed to have bacteria in it or their spores. He didn't know anything about, by the way, he didn't know anything about bacterial spores. I'm adding that because it's a fact. Pasteur didn't know anything about bacterial spores at that time. He was very lucky. I'll explain why. In this experiment here, he simply boiled the nutrient broth. He was very, very lucky because all the bacteria that were present here in the broth were actually living cells. They weren't spores. Spores are resistant to boiling. If he'd had spores in there, his experiment would have failed. But he didn't. He had just living bacteria. He was very lucky. Somebody else later on repeated his experiment, and they were unlucky enough to have spores in, and it's reinvigorated this whole debate until somebody was able to say to them, no, you show them that they had bacterial spores present. So now Pasteur says, look, I've got all the makings here in the broth. It's got free access to the vital force, and spontaneous generation should occur if that vitalist theory is true. And it never did. And it was the definitive proof. It was definitive. Spontaneous generation does not occur. It is not a real thing. Once that was established, then all sorts of aspects of biology could take off. All sorts of experiments could begin to be done, as long as this bugaboo of spontaneous generation was removed. That period, especially the advent of Pasteur, opened up an era of intense exploration in microbiology. And this is often referred to as the Golden Age of microbiology. Actually, we're in a second golden age of microbiology, spurred by our understanding of DNA and its function. But the golden age of microbiology exploded, especially with the work of Pasteur. Now, Pasteur was, he was born in 1857, in the mid-19th century. He came into a world where, for example, they had moved far beyond Van Leeuwenhoek's Hook's simple little microscope. They had developed compound microscopes, which were very much better seeing things. They could see bacteria quite easily, but they had a very, very, very poor understanding of what bacteria did and of their involvement in the natural world and everything else. They didn't actually believe, most scientists did not believe that bacteria could actually do anything. They thought they were just little small living things that just hung around and took advantage of certain situations and grew up. And for instance, they knew that you got lots of bacteria on rotting meat, and they could pull bacteria off rotting meat, but they didn't ever think that bacteria were contributing to the rotting. They just thought meat rots and bacteria grow on the results of it. They didn't ever think it caused anything. They didn't think bacteria were big enough or complicated enough to actually do anything. But Pasteur changed all of that. The very first important thing that Pasteur did for all of us was that he worked on wine. He was actually employed by vineyards. Now, the vineyards in France, of course, are extremely famous. It's a huge industry. And they had noticed that as they used to ferment their wine, and if they produced it, they would keep some of it, of the ferment. And if the wine worked very well, they'd be able to use that sample again to do the next batch of wine. They had no idea what they were doing. They simply knew that it worked. We say that's empirical. It's not, and it just worked. They knew it worked. That's what they did. If a batch of wine worked very well, they'd keep some of it and use it to seed the next lot of wine. They had no idea what was happening. They had no idea of the process, of the biological process or the chemical processes that produced wine from grape juice. They had no idea why alcohol resulted from fermentation, for example. They employed Pasteur to have a look at the winemaking process to try and help them improve these cultures that they had. And he examined them and he saw that they were very rich in yeast. And he came to believe that the fermentation process was taking grape juice and it was converting the grape juice into the wine, and most importantly, it was converting it into a different substance, into different substances which tasted different, and especially which produced the alcohol. That in itself was a revolutionary idea, that a microbe could actually do something, was something that people would argue about. But what Pasteur did was he developed ways of actually culturing those yeasts. And he treated the yeasts exactly as you would treat a group of farm animals. He actually selectively bred yeasts. He bred different strains of yeast to do different things. He characterized what those yeasts did. And this was absolutely revolutionary because it told people that these little microbes could actually do something, that what they did was very specific, and that we could actually get a handle on what they did. And above all, that microbes could be useful. Pasteur was a saint to the French winemakers. He was an absolute saint. He revolutionized the process of winemaking in France and the whole world. because from his work arose this knowledge that what we're doing is we're actually using yeasts. Cultivation of yeasts, of different yeast strains, etc., became a major thing, which the wine industries did. So that was when he was a very young man. And look at what he's, look, we've already acquired two major things. He's going to demonstrate that spontaneous generation doesn't occur. And here he is demonstrating that microbes actually do something, and that it can do something useful, that wine is produced from yeast, which, by the way, I remind you, is a fungus. Yeast is a fungus. It's not a bacterium. So he also was very interested in that whole process of fermentation. And he didn't, it was many years before the biochemistry of the fermentation process was elucidated, but he realized that there were certain conditions under which it occurred and certain conditions under which it did not occur. The yeast, for example, could live perfectly happy in the air. It could live happily in a well-aerated broth and grow really well, but it would not ferment anything. But if it was necessary to isolate the solution from air, and once you did that, fermentation would begin. So he knew that this was a process associated with anaerobic conditions, the absence of air. He also knew that what the microbes were doing was converting sugar to alcohol, because he measured how the sugar decreased in the grape juice and how the alcohol content increased. So he was able to establish the parameters of the fermentation process, even though it was many years before it was finally worked out in its detail. He looked at this and he also thought, now, what are bacteria doing? Yeasts are fungi, but what are bacteria actually doing? Can they do similar things? And one of the things he was interested in was the processes of decay and of spoilage. And the answer is yes, bacteria can do similar things. They definitely ferment things. They can produce all sorts of products like alcohols as well. He became interested in the processes of spoilage, because spoilage meant such a waste. People would produce a huge amount of food, but a huge amount of food went to waste, for example, because it rotted. And he was interested in those processes. A lot of his interest went, for example, again to the wine industry, because wine, if it's left, tends to spoil. It actually spoils because of bacterial action, and it turns to vinegar. And this meant a huge loss to the wine industry. And he studied this really carefully. He knew it was nothing to do with the yeast. And then he found that if you took wine and you heated it gently, and you kept it at a gentle heat for a long time, and then let it close the bottles and cool them, the wine did not spoil. He was able to kill the bacteria with heat, and as long as the heat was gentle enough, it would not evaporate the alcohol, and it did not radically change the taste of the wine. It changed it a bit, but it didn't radically change. It was still perfectly drinkable, and above all, it lasted. That is how we've ended up being able to drink wines from the 1800s. It was because of Pasteur. Before that, before wines were pasteurized like that, they very frequently spoiled. So that was another contribution that he made, all associated with the wine industry. Amazingly enough, I am not even sure that he drank wine. I'm not sure. He certainly never drank it heavily. With all of Pasteur's work, which we'll return to in a minute, people paid very close attention to Pasteur and to what he was doing, and they began repeating his work. they began amplifying his work. And associated with this idea of the fact that microbes could actually do something, people began to wonder, is it possible that bacteria are actually causing disease? Now, a lot of the work, initial work, was actually done empirically. People simply found methods which reduced the incidence of disease and didn't really understand the mechanisms that they were using. But here's an example. This man, Joseph Lister. Lister was a surgeon who worked in London. And I need to tell you a couple of things about the surgeons, about how they operated. Surgeons in those days, they actually did nothing to keep themselves clean. Surgeons used to wear an apron, and the master surgeons were distinguished by the fact that their aprons were filthy, were caked with blood. The more blood and everything that they had on their aprons, the more skilled they were reckoned to be. They didn't wash their hands. They went from patient to patient indiscriminately. They would amputate a gangrenous leg and then go and deliver a baby. So it was no wonder that going to hospital was an extremely risky proposition in the 1900s, with no disease control whatsoever. The mortality rates were absolutely enormous. I'll tell you a little earlier story. There was a doctor called Dr. Semmelweis in Austria. He went to a hospital where mortality rates were extremely high amongst women giving birth. This is due to what it's called post-puerperal fever. It's actually a staphylococcal infection of the reproductive tract and then a sepsis. In those days, it killed many women. Childbirth was an extremely dangerous undertaking, especially if you went to a hospital and especially if you had a doctor. It was less so if you had a midwife, because midwives spread their deliveries out. The midwife had other occupations. She was a housewife or whatever. She'd wash her hands, go and deliver a baby, come back and milk the cow, right? And it'd be 2 days before she went and delivered another. The surgeons and the doctors went from woman to woman to woman to woman to woman and amputated a leg or two in between. They acquired the germs, they acquired the bacteria, and then they spread them through the hospital. Semmelweis had an obsession with hand washing. He insisted that all his staff wash their hands regularly, just with soap and water. They had to wash their hands continuously. And he was able to reduce the mortalities in the wards where he worked spectacularly. It's actually been documented. We have the data to show how Semmelweis impacted on survival. But unfortunately, he was a very contentious figure. He was very difficult to get on with. Very few people believed that these germs that they couldn't even see could cause disease. And ultimately, he was ousted. And as soon as he was ousted, the mortality rates in his hospital rose back again. But the realization that bacteria could cause disease was actually, I'm trying to chase my cat away because it's meowing at me to be fed. This idea that germs could cause disease actually was filtering out. So here's this gentleman, Joseph Lister. He's a surgeon, but he listens to this and it makes sense to him. He's paying attention to Pasteur as well. And in 1865, in his operating room, he began to use a carbolic spray. That is phenol. It was a very nasty environment to work in. And he produced a mist of disinfectant in his operating room. He disinfected all his instruments. They washed their hands. They didn't yet use masks or gloves, but they washed their hands in phenol. And he developed these systems of he called it antisepsis because sepsis was the process whereby so many people died. And he demonstrated that if you performed antisepsis, you could improve your survival rates enormously. And his methods actually began to be promulgated, even though people didn't really understand what was happening. They just saw this actually worked. And so they began to do it. And this was, of course, because microbes would infect wounds. Microbes would infect a vagina and uterus that had just given birth and cause this incredible sepsis that resulted in the mortality. So this idea that microbes could cause disease was called the germ theory, and it excited enormous controversy. There were lots of people who said that germ theory did not apply. In fact, there are people who still say bacteria don't cause disease. But this gentleman, Robert Koch, he sits along with Pasteur as being the masters of the golden age of microbiology. And he, in actual fact, I'm sorry, he in fact is the first person to conclusively demonstrate, although many people already believed it, he is the first person to conclusively demonstrate that microbes actually do cause disease. And as a result, the whole science of microbiology, bacteriology exploded. He demonstrated that a bacterium could cause this disease called anthrax. The bacterium was actually called Bacillus anthracis, and he described it, and he described its disease process, and he positively demonstrated that it did cause disease, and he did it by performing an experiment, which is a classic experiment, and the results of this experiment are still applied today in modern science. So here's what he did. First of all, he set up a series of postulates about things that would happen if you performed this in a certain way. And we still accept these postulates as being the definite proof that a particular organism causes a particular disease. So, for example, anthrax is a disease which affects farm animals. This affects us as well very badly, but it was principally important because it could wipe out large numbers of livestock. And first of all, they're quite easy to recognize. Bacillus anthracis is quite easy to recognize under the microscope. So Koch, first of all, characterized the bacterium. He went to these animals that had died of this disease. Nobody knew why. And he found in them lots of these bacteria. And he always associated these anthrax bacilli with the death of the animals. So he then took these bacteria and he postulated that if he put them into broth, he would be able to grow them. He did it very, very carefully, and he was able to grow the bacteria. Postulate 2. Postulate 1, the bacterium, the organism must be isolated from an animal, whatever, where a disease has been caused. 2, postulate 2, you must be able to cultivate that microorganism. Postulate 3, very importantly, if you take that organism and you inject it into an animal which is not ill, that animal must acquire the same disease as the first animal. So the second animal acquires the same disease. But here's the fourth postulate, which people often forget about, and that is you must be able to extract the same bacterium from your experimental animal. So those are the 4 postulates. 1, you must recover the microorganism. You must be able to culture the microorganism. The cultured organism must be able to cause the disease, and you must be able to recover the same bacterium from the experimental animal. Those are called Koch's postulates, and they remain the standard way in which we prove that a particular microorganism causes a particular disease. Now, I just want to finish today's class at this point. We've talked a lot about the Europeans and, you know, all these rich gentlemen and all of these gentlemen scientists and everything. And fine, they're all very important, and unfortunately, you know, there are not many women scientists in there, but I'll tell you that one of the most important microbiological developments was made by a woman, and she was an American. Her name was Fanny Hess, and she was married to Koch. And here she is. And here's the difficulty. Koch could grow, they all could. They could grow bacteria in broth very, very easily. If you still do it, you'll do it in lab. You grow back, you put here, you have sterile broth, you put some bacteria in it, and the bacteria will grow. There's a big problem, and that is it's very difficult to see what you've got in here. Even if you look at them under a microscope, very often one bacterium will look like another. And it's very difficult to tell whether what you have here is a pure culture, just one kind of bacterium. And so they realized that what they needed to do was they needed to develop a system where they could have a solid surface on which they could spread their broth and see the bacteria, because when bacteria grow on a solid surface, they will grow and they'll make a colony, and the colony has its own particular characteristics, and you can sample from the colony and examine them under the microscope. It's a very useful technique. It gives you all sorts of capabilities that we'll talk about later on. They couldn't find a solidifier. They tried to use jello, but gelatin is extracted from animal carcasses. It's an animal protein. And most of the bacteria that they were working with could digest gelatin very, very easily. So they'd make a plate. They'd pour the plate with gelatin and then try and spread their bacteria on the surface of the semi-solid gelatin. And then when the bacteria grew, they just turned the whole thing to mush, and it would never work. But Fanny Hess was a good homemaker, and she used to make jams and jellies. And she did not use gelatin to solidify her jams and jellies. She used a substance which is extracted from seaweed called agar. And she gave them the idea of taking broth and adding agar to it and making a solid surface. And that's what they did. They'd sterilize it, pour it into a petri dish, solidify it, and it was a perfectly good surface on which you could put a solution of bacteria and have them grow. And you could see them growing, you could see their characteristics. Exactly how we do that, I'll discuss in a while. But it was one of the most important steps in the development of microbiology as a science, and we have Fanny Hess to thank for it. Okay, so I'm going to stop there, and we will return to this again on Friday. Okay, I'll see you then.